Cynvenio Biosystems is a startup based in Southern California in Thousand Oaks. We were founded in 2007 with some technology from the University of California at Santa Barbara. We're focused on the isolation and the identification of circulating tumor cells in whole blood. So what that means is that we take a regular blood draw from a patient. We find these cells that tend to be very, very rare, even in chronic disease, and then we sequence them. We look at their DNA. We try to determine where they've come from and where they're going. So it's a tool for doctors. It's a diagnostic. It will become a broadly adopted diagnostic. It is our hope in a few years. And we work with physicians and pharmaceutical companies. Well, that's actually the focus of the research. We work with cells that have detached from primary tumors, from metastases even. So we identify those and we look for the mutations that they're undergoing, which inform as to their future, whether they're going to form a new disease, a new tumor. But we also look at other cells that are present in the blood. Yeah, they're very rare. These cells can be as rare as 1 or 2 per billion, or as many as several thousand. The technology that we developed is very sensitive, so we find many of these. We find hundreds, if not thousands. We also tend to amplify them, so we expand the cells that we capture, and then we keep them viable so they can be, in our case, sequenced. We work with the companies that are sequencing these cells. They're building a molecular map of these diseases. So we're the input to companies that are looking at molecular structure. We use magnetic beads. These are very, very small. These are microscopic magnetic beads. They bind, they're covered with specific antibodies, and they seek out and bind to the cells that we're interested in extracting. Then we flow this substance through a chip, a special chip that we developed, and the magnetic fields are exerted externally, and they attract the beads with the cells, the targets that we're interested in. Then depending on whether we want to image them and count them, we stain them on the chip and have scanners quantify these targets, or we cleave them from the bead and we retrieve them, and then we process them downstream from the chip and other technologies. Again, primarily sequencing. We are very focused on sequencing, so you'll hear me say that a few times. Yeah, we work primarily with breast cancer, ovarian cancer, colorectal, and pancreas, although it applies to several other types of cancer. There isn't yet the clinical data. No, the cancers share certain surface markers on these cells. So we use something called EpCAM, epithelial markers, for these cells. But there are other markers that are specific to each disease state. And those are known in science, and we look for those. Yes. So normally companies are categorized as research stage, revenue stage, et cetera. We're sort of all of those things at the same time. And that has to do with how we started. We're privately funded. And we're not venture-backed, which means that to finance the development of the technology, we wanted to create some commercial products, which we did 2 years ago. We also had a contract from the U.S. Army, which we performed against and delivered for something slightly different that uses our technology called bacterial phage display. But we also have commercial contracts with some pharmaceutical companies that have provided enough funding for us to develop our technology. So we're a research stage, revenue-generating startup. And we are not venture-backed, at least not to this point. So we've been privately funded and have revenues from commercial partners. Well, I can tell you, with a lot of conviction, what I'd like to see this science become is a broad screen for the population. I'd like all of us to be able to have a regular checkup and by means of a normal blood draw, have the physician tell us, what is the future looking like? Is there a disease that is on its way before it takes hold? So a broad screen of the population is, I think, what the promise of this technology is. I think it's going to take a few years to get there. There's a lot of clinical testing that needs to be done, so that informs as to the work that we're doing now. We are working with patients that are in treatment, so we look at, we provide a window into the patient for those doctors, a molecular response that can be measured to see if the drugs they're taking are effective, or if those patients even will respond to a certain drug, and we help the doctors figure out combination therapy and the right dosage, or if the toxicology is incorrect. It's a window into the patient today. We also look for recurrence. I'm very focused on breast cancer because this is an extremely good predictor of the return of the disease, much earlier than can be imaged. So we like to give the physicians and the patient a very, very early jump on the recurrence of the disease. Then they can be treated and it can be prevented. But longer term, if I could pick one thing, I would like this to be a broad screen for wellness. And I think that's what it's going to be. And I think it's going to take 10 to 15 years.